In this video, I'd like to talk about sugars and relate them to a key molecule, right? And the molecule that I'd like to talk about in this video is glycogen. In the realm of polysaccharides, you can have either homopolysaccharides or heteropolysaccharides. Let's start with the homopolysaccharides. So these sugars have glucose, right? So these polysaccharides are made only of glucose, right? So they're essentially repeated units of glucose. Oops, sorry about that. So for example, we can have, here's one glucose molecule, here's another glucose molecule, here's another glucose molecule, here's another one. So they're all the same. That's why homopolysaccharides are made of the same monosaccharide. And the ones we'll be talking about consist of glucose, repeated glucose polymers, okay? So the first one is cellulose, which is a structural homopolysaccharide, okay? Cellulose is found in plants with beta-1,4 links, and it plays mainly a structural role in plants. Hence I drew this leaf here, because cellulose, right, we can eat it as fiber, for example, is found only in plants with beta-1,4 links in it, okay? Another type of homopolysaccharide is the form that allows plants and animals to store energy. So these can include starch in plants, and starch can be in the form of amylose or amylopectin. Amylose is not branched and amylopectin is branched. And I put P here indicating that these are found in plants in the form of starch for plants to store energy. Then you have glycogen, which is found in animals, in us, for us to store energy to use when needed. Glycogen is the most branched of the energy storing homopolysaccharides, and this allows us to easily access it for energy because it has so many bonds, right? We can access the molecule so that we can retrieve a lot of energy from it. So this branching allows us to use it and allows us to access the glycogen so that we can get a lot of energy out of it when we need it, like when we're undergoing longer fasts, for example, or when we are studying for very long periods of time, right? When we're fasting, when we're exercising a lot, when we really need to use that energy, then we can use glycogen, okay? And glycogen is very branched, right? So amylose in plants is not branched at all, and here I drew just a linear, polymer. Amylopectin in plants is slightly branched, and glycogen in animals is really branched. And all of these have an alpha 1 to 4 linkage, okay? So all of these molecules have an alpha 1 to 4, unlike the beta 1 to 4 in the structural cellulose of plants, right? Now, amylopectin and glycogen, which have the branches are actually an alpha 1 to 6 linkage, okay? So the branches are alpha 1 to 6. But the other, standard links, bonds, are actually alpha 1 to alpha 1,4 linkages in these energy storing homopolysaccharides, versus the cellulose was the beta 1 to 4. The second big group is heteropolysaccharides, which contain disaccharides, which are 2 sugar unit repeats. So these are actually disaccharide repeats. Let me actually put this down. So these are disaccharide repeats. So disaccharide means there are 2 sugar units and they repeat. And the sugars can be either glucose, galactose, or idose. And these can also have like amino groups, acid groups, or sulfate groups on them. These are not branched, have variable links, okay, and play a structural role in animals. So like glycogen played like an energy storing role in animals. These guys play a structural role in animals. An example is GAG, G-A-G, okay, and this stands for glycosaminoglycan, and you can find these in places where you need extra structure. So these GAGs, heteropolysaccharides, can be found in the eye, in the vitreous humor, for example, and in the cornea, in tendons, in cartilage, in the skin, in the synovial fluids of joints, etc. So they provide structure for animals, okay? And they're not branched, they're just linear polymers of these 3 sugars, right? So, I mean, they're a linear polymer. They can contain one of these 3 sugars, and they're disaccharide repeats. So they're a repeat of 2 of these 3 sugars, depending on what GAG we're talking about. Okay, there's different kinds, but they can have any of these 3 sugars, and they're going to be disaccharide repeats, which are not branched, and they provide structural support. They play a structural role in animals. So these are found in animals. These GAGs are found in animals. Okay, but the key for us animals is glycogen to make energy, right? Sorry, to store energy. That is, to store energy so that we can later use it. And the GAGs, which play a structural role. So we will talk about glycogen later when we look at glycogenesis and glycogenolysis. So glycogenesis is making glycogen, and glycogenolysis is breaking glycogen so that we can use it for energy, so that we can make energy out of it. We have to break it down, right, to give us the energy when we need it. For example, the liver can break glycogen to glucose to make sure that the rest of your body, especially your brain, has enough of it. For example, when you study or when you think a lot, like when you study a lot or when you have to think, especially when you have been without food for a very long time, right? So the liver is going to liberate that glycogen to the rest of the body, like to the brain, for example. Your muscles can also liberate glucose from glycogen when you need quick bouts or bursts of energy. Like when you have to run to catch a bus, for example. When you have to catch a bus, when you have to run very, very quickly to the bus stop so that you catch the bus, that's a very, very quick burst of energy that your muscles need, right? They need to have the energy to perform that function. So they can also liberate glucose from glycogen and use that glucose in glycolysis to make ATP, to make energy. Okay? I hope this video was helpful, and in the next video we'll talk about glycogenesis and glycogenolysis, specifically how we make glycogen and how we break glycogen so that we can get the energy we need when we need it.